This is Dr. Liotta, and I'm really delighted to join our Mason Korea colleagues. I'm going to be talking about the application of nanomaterials to diagnostics and drug discovery. And I'm located in the Center for Applied Proteomics and Molecular Medicine, College of Science, George Mason University, and the Manassas campus, or the biotech campus. We're at the Institute for the Biohealth Innovation, and we bridge research across disciplines to address global challenges. We combine bioengineering, biomathematics, molecular biology, microbiology, and our specialty is proteomics. Our areas of focus are protein drug targets that are active in driving disease processes. So we study disease processes, and then we take our discoveries all the way to clinical testing and clinical trial examination of the efficacy in patients for personalized medicine through our CAP-CLIA certified lab. We develop and invent new technologies and apply them to biomedical research, and we've spun out several companies based on our Mason inventions developed under NIH funding. Today I'm going to tell you about some humble organic dyes that we've discovered can be used for affinity capture of biomolecules, and that could be useful for drug discovery and for early diagnostics for many types of diseases, virtually any type of disease. So these humble dyes that exist in fabrics, for fabric dyes, for printing dyes, even for 3D coloring, for 3D printing. These dyes bind to molecules with very high affinity. That's why they don't come off in the washing machine. I'm going to tell you how we're using these dyes to capture biomolecules for diagnostics or to paint the surface of interacting proteins for drug discovery. So if we think about the field of biomarkers, one of the biggest roadblocks to discovery of new biomarkers for disease is that the biomarkers coming from the diseased tissue are present in only very small amounts, and then they're diluted in the entire blood volume, and they're fragments, otherwise they wouldn't end up in the bloodstream. They wouldn't be able to penetrate the blood vessels of the tissue. And so the final estimated biomarker concentration from an early stage cancer, an early stage infectious disease is 0.1 to 10 picograms per mil for markers coming directly from the diseased tissue. This is way below the sensitivity of discovery platforms such as mass spectrometry. So how do we solve this problem? Well, Dr. Lucchini and our group developed a new type of nanoparticle that has 4 independent functions within minutes in one step in solution. Sequestration, molecular size sieving, and affinity capture using those dyes I mentioned of an analyte, even if it is bound to a carrier protein. Concentrate all solution phase target molecules, complete protection of the harvested proteins from enzymatic degradation, and exclusion of albumin. The way it works is the affinity dye is inside of a meshwork, high surface area nanoparticle, and when the biomarker of interest enters the pores of that nanoparticle, it binds tightly, doesn't come off. All the biomarkers in solution are swept out of solution, and they end up being in the nanoparticle, which we can then concentrate in a small volume compared to the starting volume. So we have a 100,000 or 1,000-fold increase in concentration of the biomarker of interest. And here you can see the capture dyes and how rapidly they bind with very high affinity to classes of proteins. This takes any assay that we want to use it for, any type of molecular assay, PCR test, antibody test, mass spectrometry test, metabolomics, glycomics. It causes that captured antigen or analyte to be cleaned up and then enhanced in concentration. So it increases the sensitivity many orders of magnitude. So now we can reach down into the low abundance proteins that were never before possible. Let me show you how this applies to tuberculosis. Tuberculosis is not a disease of the past, as it is, we think about it in the United States as a disease of the past. It is a disease of the present. 1 million children become ill with TB each year, with a 40% mortality if the mother has TB. 10 million, new cases of TB in 2016. It's continued to go up, and it's a worldwide epidemic. It's just not as bad in the United States. That doesn't mean it's any less important. So we used our nanoparticles in collaboration with Johns Hopkins University, Bob Gilman, whose group collected hundreds and hundreds of specimens of active TB from underdeveloped countries. And what we found is that our nanoparticles could capture very rapidly and concentrate, this is never before possible, a whole series of markers that come from the surface of TB organisms and also can capture the inflammatory reaction that the host has to those markers. When we apply this to study early diagnosis of active TB, we were able to diagnose HIV-negative pulmonary TB and discover previously unknown TB-specific proteins in the urine of patients with microbiologically, that's culture-positive, proved pulmonary TB with very high statistical significance, very high sensitivity, and compared to diseased controls or healthy controls with a high sensitivity and specificity. And the level of this marker correlated with cough and cachexia. And this marker has been confirmed and proven by many other investigators. Importantly, we were able to show that this diagnostic test worked across many countries, Peru, Venezuela, Guinea-Bissau, Uganda, and the United States, for active TB patients. This is a urine test, very user-friendly. So we achieved WHO criteria and sensitivity and specificity blinded across all of these countries. We also used mass spectrometry and found not only the specific sugar marker that I mentioned for TB, but we found 61 novel TB-specific, derived from TB organism proteins that might serve as new biomarkers or new vaccine targets. And in particular, it's never been possible to diagnose active TB in children by a simple urine test. We were able to do that with our test because we used urine because children cannot produce sputum easily for standard testing. And in children, TB spreads throughout the body, and it's not just confined to the lungs. And too often, diagnosis of TB in children is made at autopsy. So it was a really important problem. And we found very high sensitivity and specificity for a specific branching sugar structure that was shed by the TB organism into the urine, passed through the glomerular filtration, so it came from the general circulation and entered the urine. So that shows you how we can use the affinity capture dyes that I mentioned in the beginning for making a new, completely novel, worldwide applicable test for active TB, so that we can treat those individuals before they spread the disease. Next, I'm going to tell you about using these same affinity dyes for studying protein-protein interactions. Protein-protein interactions are the drug targets of the future. They were thought to be undruggable because they often are large interaction area, lack of catalytic sites, and they're hard to target with small molecules. Current methods to identify protein-protein interactions are very laborious, and they lack adequate sensitivity and specificity. Nevertheless, protein-protein interactions occur throughout all signal pathways at many levels in the cell and are a very important drug target area for the future. We focused on hotspots residues, which are largely contribute to the binding affinity. They act cooperatively and tend not to be distributed evenly over the surface. So how can we identify those as targets for new drugs? We used our new technology we created called protein painting, in which these novel dyes that I mentioned are used to paint the whole surface of the interacting protein complex. Then we denature the complex, digest it with trypsin, and trypsin will only cut the residues that were exposed on the surface of the interacting molecules, but not covered with the dyes. And then we just plug that right into mass spectrometry, and the output is only the regions where the proteins are binding together. We get the amino acid sequence of the three-dimensional contact points between the two interacting proteins with the resolution of 3 amino acids. It's a very simple protocol, only takes a few minutes to do. We preform the complex, could be mixtures of proteins, cell lysates. We mix them with our special dyes. We just remove the unbound dye by gel filtration. It takes 1 minute. We gently denature with urea, digest, and put it right in a mass spec. And the only thing that pops out is the interaction area. And we see by these studies, here's a dye that binds really rapidly to the protein. The number of binding sites is proportional to the protein size because it coats the whole surface. And even if we denature, reduce, and alkylate, the proteins are still covered with the dyes. And we know that these dyes bind by having a mixture of aerial anchors at the opposite ends, which secure high affinity binding by multivalent attachment to the protein. And the dyes bind without changing the native protein structure and stabilize in the presence of urea denaturization, actually. So we've applied this to same method to find a new inhibitor for osteoarthritis. It's different than autoimmune arthritis. This is a firestorm of IL-33 and IL-1 that act as a positive feedback loop that causes degradation of the cartilage and more inflammation, more death of the cells in the joint. And so what we did is identify hotspots that exist at the intersection between the accessory protein, which binds to the receptor ligand complex. And without that accessory ligand combining with the other two proteins to form a 3-protein complex, you don't get any signaling, don't get inflammation. So we used our protein painting to discover a specific set of hotspots, and we made a protein drug that would block that hotspot, and it had high sensitivity, had very good dose response curve, and we were able to have it work in animal models where it blocked the inflammation in the joints of mice. We've also used our method to discover new targets in the BRCA1 gene that's involved in breast cancer, involved in stabilization of genetic instability or genetic DNA repair. And we found that we could find phospholipeptides that mimics that are bind to the BRCA1, stabilize its activity, and we think the applications for this is to stimulate the activity of BRCA1 in a way that allows us to combine this with a PARP inhibitor for increased PARP sensitization for cancer patients. Also applied this to the Hippo pathway, which is involved in cell-cell interactions, cell growth, wound healing, and has a whole series of positive and negative interaction components to this pathway. We were able to use our protein painting to discover 3 candidate hotspots. And this was not possible before because crystal structure wasn't known for these proteins. And we found a series of different domains that are all now targets, potential targets for new peptides, new drugs that can, in particular, be useful for improving wound healing. And lastly, our last example is a new inhibitor for PD-1, PD-L1. As you know, PD-1 is a T-cell receptor that recognizes PD-L1 on the tumor cells, and when the tumor cells bind to the immune cell, it suppresses or kills the immune cell. Blocking this interaction with antibody drugs is a huge field, $6.2 billion in 2017, and now it's 10 billion or more. The next generation of these inhibitors are small molecules or peptide mimetics, and that's what we've developed. A peptide mimetic that interferes with the binding of PD-1, PD-L1. They're pretty small proteins in the first place. And here's our inhibitor, and it has very good dose response curve. And the latest version of it, which is cyclized, is very stable in the bloodstream. And we know that there's a relationship between the antibodies that bind to PD-L1. Let's look at this gigantic antibody here, the small PD-1. But our inhibitor is a small molecule, gets right in at the exact hotspot of interaction. And we think it's going to be very useful for the future for new classes of immunotherapy. So in conclusion, our center applies proteomics to a variety of different clinically important problems for cancer, infectious diseases, and neurodegenerative diseases. And I'd like to acknowledge our great collaborators and our team members of our group and the funding sources that we've been so fortunate to receive. Thank you very much.